Hi, I'm Nick Panasic, and I'm going to briefly go over how to read a scientific paper. It's something that we all have to learn how to do, and sometimes it can be quite difficult. I know when I've been teaching in class and assigned a paper, and we come to class in the morning, and I'll ask how many people have read the paper. A lot of times, I only see half the hands go up, maybe sometimes even less. And I think that's a byproduct of how difficult it is. A lot of times you start reading it and you're kind of muddling through, you don't see where it's going. It often uses a lot of jargon terms that you may not know if it's your first time reading in this particular scientific area. So I'll briefly go over what I found to be a couple handy tips. I'll use one paper as an example. Let me go ahead and I'll let you see that paper. So this is a paper uh, on the directed evolution of beta-glucosidase from Pentobacillus polymyxa to thermal resistance. So it's a directed evolution paper. And one of the questions I ask in class all the time, once a bunch of folks say they haven't read the paper, and I ask them about it, and they talk about the difficulty. I ask if you only had a few minutes to read the paper, like say you just remember before class, oh shoot, I'm supposed to read this paper, starts in 20 minutes. What should you do? What should you read? And when I ask that question, I get a lot of interesting answers. I'll get the introduction, and I think reading the introduction is very helpful. It kind of gives you an idea why they're doing the paper. It kind of sets the stage for what the research is. But then, if you only got a limited period of time, it'll take a little while to chug through that introduction. Um, some folks will say the results. I'm like, great, but you've seen the results, but you don't know why they're doing it, really how they're doing it without reading the materials and methods. Some folks will say, well, read the materials and methods. If you read the materials and methods, you don't know the results, right? Um, some folks will say, read the conclusions. And that's great if you want to know what they drew out of the paper, but it gives you no basis to really critically think about, do you believe their conclusions? A really important aspect of reading scientific papers is the critical analysis. Do you believe what they're saying? What's the proof that they use? What's the actual data that they're working with? Again, that leads you back to the results. What I do if I have a short period of time, um, and what I'll do before I actually read the whole paper is I'll flip through before I read any of this. I might read the abstract real quick, but that's really short. It's usually just a paragraph. It gives a great kind of summary. But then I'll flip over to the figures. Start with the figures. Before you read the paper, start with the figures. So we'll go to figure one here. And even if you know nothing about the topic, what this shows you is what they did and what a result was. So a figure legend should have enough information so you get kind of an idea for what the experiment was. So in this figure legend, thermal inactivation of BGLA in mutants. Okay, so they're trying to use heat to inactivate this enzyme plus some of its mutants. It says time course of irreversible thermal inactivation at 50 degrees Celsius. So what's that mean? A lot of times you'll have to kind of piece through what the words mean. If you don't know a particular term, go ahead and look it up. But you want to be able to make it through these legends. So irreversible thermal inactivation, that's just kind of a fancy way. Irreversible means there's no going back. Thermal inactivation just means you heated it up until it fell apart. And since it's a protein, this is the process of denaturation. And so they're looking at 50 degrees, and they say for wild type BGLA, that's obviously one of the enzymes they're working with, and then these different mutants. Now, if you ever see a mutant listed as like T385A, what does that mean? So what this is telling you is this amino acid, the T amino acid, and you need to understand mutations. You really need to know your amino acids. That's why we learn this in biochemistry. So T stands for threonine, A stands for alanine. So this is a threonine to alanine mutation at position 385. It's a 385th amino acid, which used to be a T, now becomes an A in this mutation. Here's an N41S, M416I. So these are all different mutants, and they tell you exactly what the different mutation is. E96K, it's an E at position 96 that's being mutated to a K. So glutamate to a lysine. And when you're looking at mutations like this, it's helpful to know your amino acids and to know those structures. That's why you'll learn them in biochemistry, because then you'll have an idea what changes are going on in the functional groups. Like for E96K, E is glutamate, which is negatively charged, K is lysine, which is positively charged. So at this position 96, you're going from a negative charge to a positive charge. Remember, charge-charge interactions are the most powerful of the non-covalent forces. So that's probably going to have a significant effect. So they go through the names of all these different mutants. Enzyme aliquots were diluted in a pre-warmed assay buffer. Samples were taken at different times and assayed for activity. So they're looking for the activity of this enzyme. They're going to assay it at different times and look to see how much activity. And so you can see this graph that they include here. On the x-axis, this is the time. And on the y-axis, they say the natural log of activity, but basically it's a percentage of activity. And so what they're looking at here, and this is what you can tell from these figures, is what did they change and what did they look at. The x-axis is always what they change. The y-axis is kind of what they measure. So after 10 minutes, and for this particular one, for this dot, which is BGLA, which is wild type, they found that it had an activity between about 1 and 2. And you can see kind of the slope of this line. So what they're finding with these different mutants, okay, so wild type starts at time 0, has lots of activity, and after 15 minutes, it's pretty much dead. Then this mutant here starts with the same amount of activity, and then after 20 minutes, it's dead. This mutant is dead at 30. This mutant is dead at 40. This mutant never quite dies over the course of incubation time, 60 minutes. And then this mutant over here, this one really isn't being affected. Okay, that's this triangle guy, which is N437K. So just by looking at this figure, you're able to tell, okay, they're comparing a bunch of different mutant enzymes to a wild type enzyme. They incubated the enzyme and the wild type, or the wild type and the mutants at 50 degrees for a certain period of time. And they saw that while wild type pretty much lost its activity fairly quickly, it denatured, this mutant up here, this N437K, seems to have really held on to its shape and maintained its activity. Just by looking at that figure, now you have something to talk about. You've seen the actual data, and who knows what conclusions they drew in their conclusion section. They may have said, well, all the mutations showed increases of thermostability. I don't know about this one or that one. Here you can make the judgment for yourself. So that's the first figure. Go through. Second figure, okay. So is this table 2, kinetic constants of wild type and mutant beta-galactosidases. So here they've got their wild type enzyme, a bunch of their different mutants. Some of these are combined mutants, so they have more than one mutation. This one looks like it has 4 or 5. And then they have the kinetic constants Km, Kcat, and then Kcat over Km they put here for you. So what's useful about this, now you know that they measured the kinetic constants and they compared their wild type kinetic constants with those of the other mutants. Again, you're getting a sense for what did they do in this paper, okay? Then we'll come over here to figure 3. This is the alanine 385, so alanine at position 385, the environment around it, it's showing you a little picture from their crystal structure. so you can see what other amino acids are around it. A detail of the molecular structure of BGLA showing the cavity around ALA 385. This position is located at the inner surface of a helix in the barrel, surrounded by hydrophobic residues, which are colored in green. Non-hydrophobic residues colored in gray are oriented towards the solvent. And so right now, not only have they compared the kinetic constants and the thermostability of these different mutants, now they're looking at a particular mutant and looking at the cavity around it. And so now they're trying to do a structure function relationship with it. And what you conclude just from this picture is they think that the cavities surrounding this mutation may have some important role. Alanine is a hydrophobic residue being buried in the midst of other hydrophobic residues is probably stabilizing. If you put a hydrophilic residue in there, it'd probably be destabilizing. So a mutation going to an ALA or something bigger that could fill that cavity that's hydrophobic may be the reason why it's stabilizing. But again, by looking at the figures, you can make that judgment yourself. And that's where your critical thinking comes in. For table 3, this is the TM, or the melting temperature, at different temperatures for wild type and their mutants. And TM is the incubation time of the enzyme at a given temperature, which results in 50% thermal inactivation. So in other words, half of it dies. So this is a measure of how quickly the stuff is denaturing. And so they have 3 columns here: 50 degrees, 60 degrees, and 65. So wild type lasts for 2 minutes at 50 degrees, and it has no activity. It doesn't last at all at these other temperatures. Whereas you start looking down further along these mutants, like N437H, that's going to last for 15 minutes at 50 degrees, 3 minutes at 60, and 0 minutes at 65. And then when you combine all those mutations, it lasts greater than 90. That's probably where they had to cut off. They didn't measure anymore after that. Same thing at 60 degrees, and at 65 degrees lasted 12 minutes. None of the others came close. So again, you get to critically analyze what you think the significance of their research is and their experiments. You can tell what they did by just looking at the figures and reading the figure legends. And that can be really, really helpful. Here now they're looking at thermal inactivation of this evolved enzyme, and they're doing a time course at 60 degrees for their multiple mutants. Okay. Samples were taken at different times and assayed for activity, kind of like what we saw before. And so you can see, when they start combining these things, they got this guy here, which is this mutant that has all these different mutations all up here. And so now you can get a general idea what their conclusion is. They got a really cool mutant that has a significantly different activity than wild type. And it seems to be very thermostable. And so you can go through in each of these pictures and kind of get an idea of what it was they did, what they're comparing, what they're analyzing, what they're changing, and then in some of these cases, even what conclusions they're drawing and why they're drawing them. Once I've gone through all the pictures, if I still have time, if I'm not in a rush, or if I'm planning on reading this paper, I'll still go look at the figures first. Now I'll go back, after seeing that, now I'll go back and read the introduction and the experimental procedures and go through the paper. I have an idea of where it's going now, right? I have a really good idea of where they're going to go with it, what kind of things they're going to be doing. And so now when I'm reading like the experimental procedures or the materials and methods, it's going to make a lot more sense to me because I know how it's going to be used and how it's going to be looked at and where this is leading to. And so now when I read materials and methods or experimental design, I can think critically. Well, okay, so they, are assaying this at a particular temperature. Did they just add the enzyme into a tube that was already at that temperature? Or did they add the enzyme to a tube and then put it at that temperature? So it took like 20 minutes to get to the right temperature. what happened during that time. I can think critically about what they did. So read your figures first, then go through the rest of the paper. Thanks.